Hello, all, and welcome to the lecture about T cell development and thymic selection. As a reminder to all of you, but you probably already know by now if you've been viewing the other lectures in this lecture series, you can follow along with your textbook. Any figures that are taken from the Janeway textbook will be referred to on the slide by number in the upper or lower corner. And this will be particularly useful as some of the writing on those particular figures can be a little bit small. So starting off this lecture, I want to talk about primary objectives, which are the objectives that we hope to learn over the course of this lecture. So first, we want to learn the stages of T cell development, starting in the thymus, and then, starting in the bone marrow, and then moving to the thymus, and then exiting into circulation. Second, we want to understand the processes referred to as negative and positive selection and why they are particularly important in T cell development. So we will go through how they happen and why they matter. And lastly, we want to distinguish T cell development from B cell development, comparing and contrasting the similarities, which there are many, and looking at their differences. So first, let's just start out with an overall view of T cell development, starting from the hematopoietic stem cell stage, or HSC. As we have already covered, these cells give rise to all different cell types in the blood, including the myeloid compartment. But today we are specifically focusing on the development through the lymphoid compartment. So this will go through the common lymphoid progenitor, also referred to as CLP. And then today, we are specifically focusing on T cells, which derive from these common lymphoid progenitor cells. And so they will start out in the bone marrow, then they will move to the thymus to undergo most of their developmental stages. And these cells will also give rise to effector functions, which are the actual function that they take on during the immune response. And this will largely be covered in subsequent lectures. Today, we're primarily focusing on the stages of development that start at the hematopoietic stem cell stage, moving through the bone marrow to the thymus and undergoing the development primarily within the thymus. So first, I wanted to start about talking about the organization of the thymus. The thymus is located right above the heart, just to contextualize where it is in the body. And within the thymus, there's specific organization of different cellular compartments. The top part being referred to as the cortex or the cortical region, and the bottom part being referred to as the medulla or the medullary region. And different stages of T cell development will occur at different sites within the thymus, which is why it's important to understand these different sites of the thymus, and we will come back to these different locations at which T cell development occurs at the end in our summary. But the T cells, or the cells that will become T cells, enter through the medullary region of the thymus, move up through the cortical region during key steps of development, before moving back to the medullary region in which they conclude development. So giving a general overview of the life cycle of a T cell, as I've mentioned several times already, the T cells, like the B cells, start out in the bone marrow with these progenitor lymphocytes. But before continuing on in T cell development, they will move from the bone marrow through circulation to the thymus. And then when these progenitor cells reach the thymus, they will receive these key signals, which we'll discuss further, in order to commit to a T cell lineage. There are also other important stages within the thymus, such as negative selection, which will eliminate autoreactive T cells. This process occurs a little differently than what we have learned about during B cell development. However, it is a similar general concept that we are trying to eliminate our autoreactive T cells. Before getting into the specific stages of T cell development, it is important to understand that there are different types of T cells, and that will change the trajectory at which under which they go during development. So there are two major types. The first type are alpha-beta T cells, and this alpha and beta just refer to the T cell receptor chains. There's an alpha receptor chain and a beta receptor chain, and these types of cells can be subdivided even further depending on the co-receptor that they have. So there are two different types of alpha-beta T cells, CD8 positive. These have the CD8 co-receptor, which, if you remember from our antigen presentation lecture previously, this co-receptor facilitates interaction of this alpha-beta T cell with MHC class I when it recognizes its antigen. Conversely, there are also CD4 positive alpha-beta T cells, and these will interact with their cognate antigen through MHC class II. The other major type of T cell are these gamma delta T cells. So they differ from the alpha beta T cells in that they have a gamma chain and a delta chain. However, alpha beta T cells are what we are primarily going to focus on during this lecture because they are more well understood and they are the more common type of T cell. But before we get into those stages, let's talk about these T cell receptors a little bit more. So over here, we can see our beta and our gamma chain are organized in a very similar fashion to that of our B cell receptor heavy chain, in that they have V, D, and J segments that will all undergo recombination in order to form a successful beta or gamma chain. By contrast, when looking at our alpha and delta chains, these just consist of the V segments and the J segments, so this is more analogous to the light chain of the B cell receptor. So as we did with B cell development, I want to talk about the major accomplishments that we are trying to achieve during T cell development. So this first, and as mentioned before, this is primarily referring to those alpha-beta T cells. So first, we will undergo rearrangement of the beta chain. Next, we will undergo rearrangement of the alpha chain. Then there are these rigorous selection processes in which the T cell receptors are being selected on their ability to interact with either MHC class I or MHC class II, and this is referred to as positive selection. And then there's another selective process by which T cells undergo so that they are eliminated if they are autoreactive. And this is also referred to negative selection, kind of like the negative selection that is undergone during B cell development. So here is just an overview of all the stages of T cell development. And I just wanted to point this out right here. These particular cell type markers are useful for immunologists to distinguish these particular cell types. They're not necessarily important functionally, but oftentimes immunologists will try and determine different cell types from each other and these different cellular stages. So they're just useful markers during experimentation to look at these different cell types. So we start out in the bone marrow, and then we move to the thymus, under which this double negative stage will occur. And this is a stage in which the cells do not express either co-receptor CD4 or CD8. Then the T cells will move to this double positive stage in which they express both CD4 and CD8 co-receptors. And then before maturation, they will start to express only a singular co-receptor, either CD4 or CD8. So let's look at these in a little bit more depth. Starting with our first double negative stage, referred to as Dn1. This is the stage at which the T cells, the precursor cell has moved from the bone marrow into the thymus, and these particular thymocytes will commit to the T cell lineage. The way that this occurs is once these precursor cells enter into the thymus, they interact with these thymic stromal cells that express these particular notch ligands on their surface. So these precursor cells have these notch receptors on their surface, which results in signaling to the T cell and ultimate commitment of that cell to the T cell lineage. Then we move to the double negative 2 stage after committing to T cell development. And this is the stage at which RAG expression will increase, and so the TCR beta rearrangement can proceed. Okay, so at this point, we are going to walk through the path towards beta chain rearrangement. So here we start with our double negative thymocyte, meaning it doesn't express either co-receptor. And during the Dn1 stage, this is when we increase RAG expression in preparation for VDJ recombination that is necessary to recombine the beta chain. Then we move to the Dn2 stage, at which both the D to J betas on both alleles are undergoing rearrangement. Then we move to the Dn3 stage. Following the D to J beta rearrangement, we have the V to DJ beta rearrangement at a singular allele first. And if this recombination event is successful, then, much like during B cell development, we have allelic exclusion, where this particular chain will be epigenetically inaccessible to the recombination machinery so that it will not undergo recombination. And then, if this is unsuccessful, we will proceed with recombination at the second allele. If this recombination event at the second allele is successful, then the cell will also be able to move on to the next stage of development. And if this is unsuccessful, the T cell will not be able to have a beta chain, and so it will undergo cellular death. So for these cells that have a proper beta chain, what is next in their cellular development? This is when we move on to the Dn3 stage, which is very similar to the pre-B cell stage, in which we have a pre-T cell receptor that's expressed. And this is necessary to tell us that there is a successful beta chain to signal to the cell that it can move on because the beta chain has rearranged successfully. So much like the BCR heavy chain rearrangement, in which it was paired with the surrogate light chain and there was this mimicking of an antigen recognition response, which resulted in receptor signaling to the cell to tell it to move on to the next cellular stage, the T cell receptor at this particular stage will signal to the T cell if there's proper beta chain rearrangement. So over here you can see a T cell receptor, and it looks very similar to that of the pre-T cell receptor, the difference being that instead of having your alpha chain, you have this pre-T alpha, also referred to as PT alpha for short, and this is a lot like the surrogate light chain for B cell receptor development, in which it can signal to the cell to undergo allelic exclusion so that it stops recombining its beta chain and doesn't recombine its other beta chain on the other allele. And so it also will signal to the cell to undergo proliferation, much like with the pre-B cell receptor signaling. If you remember from our lecture on B cell development, these receptors are not actually able to leave the ER and move to the cell surface unless they have been paired with their cognate chain. And this is no different for T cell receptors. So normally, before a T cell receptor is able to move to the cell surface, the alpha and beta chain need to be paired together. This provides stability to the protein and allows it to no longer be retained within the ER and move to the cell surface. This process in this particular Dn3 stage is very similar, but instead of the alpha receptor, because the alpha receptor has not yet been recombined, we have this pre-T alpha, which is the pre-T cell receptor. And once these pair together, they can move to the cell surface, and this will allow for downstream signaling through that receptor, telling the T cell that proper beta chain rearrangement has occurred and that it can move on to the next cellular stage. So this cell surface expression is what tells us that we have this stable, viable T cell receptor. At which point, this cell gets a signal to undergo proliferation. Because it's signaling through the particular T cell receptor, this will also happen when a T cell recognizes its antigen. So it's very much mimicking antigen recognition with this pre-T cell receptor. Once again, because of this proliferation effect, we have the chain pairing diversity, in which we have many cells that have all undergone the same recombination event to result in beta chains that are the same, and then subsequently the TCR alpha rearrangement will occur. And so this one beta chain is capable of giving many different types of antigen specificity by having different chain pairing of that beta chain with different alpha chains within these different cells. So at this point, after we've had this signaling and proliferation, the cells will start to express both of the co-receptors. So we're moving from the double negative stage, in which neither of the co-receptors are expressed, into the double positive stage, in which both CD4 and CD8 are expressed on the cell surface of these developing T cells. At this particular stage, the double positive stage is where we begin rearrangement of our alpha chain. And if you remember looking at how the locus is arranged, we just have our V segments and our J segments. So there's only one recombination event that will happen between the V and the J, unlike the beta chain, which had to undergo D to J rearrangement and then V to DJ. So an initial attempt at V to J arrangement of the alpha chain will occur. And if this is unsuccessful, then as mentioned previously, if there are extra variable segments, this can undergo successive recombination until there are no more V chains available for recombination. So it will try and undergo successive rearrangement, hoping for a proper alpha chain rearrangement.